your purines, pyrimidines, the DNA synthesis, and the replication process. Now we will be talking about the RNA synthesis in the form of transcription, the processing and modification after. This is very important because whatever you see in yourself right now is a part or is a result of your DNA replication and RNA transcription and translation. Every bit of protein in your body is a product of your RNA transcription and translation. Okay, so for first we'll talk about chapter 36. Now, we've talked about this last Monday that we have different kinds of RNAs, prokaryotic or eukaryotic RNAs, marami sila. But please remember that there's only one who codes for a protein. That is your messenger RNA. Okay? Messenger RNA is the template of your DNA that codes for protein, no other else. Okay? Mapa-ribosomal RNA, transfer RNA, silencing RNAs man yan, wala silang they don't take part in coding for protein. It is only the messenger RNA. Probably, ribosomal RNA and transfer RNA takes part on protein synthesis, but they do not copy or they do not transcribe DNA sequences, only messenger RNA. Now, how is your RNA different from DNA synthesis? So, first and foremost, the most important is ribonucleotides are used rather than deoxyribonucleotides. So, hindi siya dumadaan sa reductase mong dalawa. Okay? Yung materials mo hindi dumadaan sa dalawang mong reductase na diniscuss natin last time. Okay? Second, another important difference is that uracil is used rather than thymidine as the complementary base for adenine. Okay? Wala kang makikita sa RNA mo na thymidine. Okay? Pag may mag-exam tayo, tapos maglakay ka ng thymidine sa RNA, automatic 0 na yan. Okay? It is only the uracil that is seen in your RNAs, never thymidine. Third, primer is not involved. Remember the DNA replication procedure wherein you'd still attach an RNA primer first before the DNA polymerase can copy your other DNA template. Dito hindi mo na kailangan, ha? Kasi RNA na siya kaagad, okay? So, you don't need a primer in RNA synthesis compared to DNA synthesis that you need a RNA primer. Fourth, only a portion of the genome is needed to be transcribed or copied into RNA. We've talked about this as well, your active and inactive zones. Those are the ones that code for your protein and those ones na hindi nagko-code for protein. Pero sa DNA synthesis, kailangan sila kopyahin lahat. Okay? Sa DNA synthesis, purine per purine, pyrimidine per pyrimidine kinokopya. Pero sa RNA synthesis, meron lang part ng genome mo na kailangan kopyahin para makaproduce ng protein. Okay? Fifth and last, there is no highly active or efficient proofreading in an RNA transcription. Why? Okay, compared to DNA synthesis, meron tayong tatlo. We have the DNA polymerase 1, 2, and 3. Given that DNA polymerase 3 is the one that elongates and adds the deoxynucleotides, while that DNA polymerase 1 and 2 proofreads. Dito walang protein na gumagawa or enzyme na gumagawa ng proofreading dito. Okay? Sa RNA transcription mo. So please remember this 5. This is a very important concept. Now, let's talk about RNA transcription per se. The sequence of ribonucleotides produced is complementary to the sequence of the one strand of the sDNA. Remember, only one strand is needed. Unlike DNA replication, dalawa silang kinokopya talaga. Okay? And after DNA replication, the resulting the double helix, the two double helix has the new strand and the old strand. Dito walang new strand, old strand. Ang meron lang RNA, messenger RNA, okay? Or an RNA transcript. Now, directions tayo. We have the 5' to 3', the hydroxy and the phosphate directions. Please remember that you code RNA 5' to 3'. So ang lalabas na RNA transcript mo is from 5' to 3' direction. Okay? Please remember, ang lalabas na ano dito, ang lalabas na RNA template mo is 5' to 3'. So, the one that you are coding na DNA template strand is 3' to 5'. Ito siya. This one. Okay, 3' to 5'. Yan yung template strand mo. Okay, your 3' to 5' is your template strand. While your coding strand, 5' to 3', yan yung coding strand mo. Huwag kayong malilito dito kasi lilito-litohin kayo ni Dr. Monsanto dito. Your coding strand is identical to your messenger RNA or RNA transcript. Kaya siya tinawag na coding strand. Siya yung gusto mong makopya. Now, paano mo siya makokopya given the fact na by base pairing tayo? You use a template. Your template is the partner of your coding strand. So kung ano man yung lumabas na messenger RNA or RNA template dito, kapareho ng coding strand mo. Kaya siya tinawag na coding strand kasi siya yung kokopyahin mo. Pero para ka makakopya dito, kailangan mo ng template strand. Okay? Parang middleman lang ba kung kumbaga. Anyway, everybody during med school, meron talaga yan. Template, may template, may coding, meron kang kumukopya talaga sa klase. So, para ka makakopya dun sa coding strand, kailangan mo ng middleman. That is your template strand. Okay? Siya muna yung pa-partner dun sa coding, tapos dun ka nakukopya sa template. So, look at this one. Remember this? The third rule, di ba? Walang thymidine sa RNA. So, you get, tingnan mo, pareho lang sila ng coding strand. Adenine, adenine. Thymidine, wala naman tayong thymidine sa RNA, so papalitan siya ng uracil. Tapos, guanosine, guanosine, adenine, adenine, cytosine, cytosine. Look at the template strand, okay? Hindi siya pareho kasi siya lang yung pinagbasihan ng kokopyahan mo. Okay, siya yung pinagbasihan pero yung kinokopya mo talaga yung coding strand. And the direction, 5' to 3' ng RNA strand mo ha. Remember, RNA strand 5' to 3' while your template strand codes 3' to 5'. Hopefully na ano ninyo to, na remember. This is the generalities of your RNA transcription. Kaso, ewan ko sa mga brilliant na tao, gusto nila ng deeper knowledge. Kaya meron tayong tinatawag na promoter. Sino si promoter? Remember, promoter is not a protein. It is a DNA sequence. Not an RNA sequence, not a protein, but a DNA sequence. Okay? It is a specific site in your DNA template strand na magsasabi na dito ka magsimula. Okay? It is composed by your consensus and your data box. Remember this two ha, consensus and data box. Kasi lilito-litohin kayo ni Dr. Monsanto. Data box, cut box, cut box. mga box na yan. Tatandaan ninyo kung saan yan sila, where do they take part. Okay? Your tata box, this one, tingin sa baba, yan. Kaya siya tinawag na tata box. T-A-T-A-A-T-A-T-A-T-A-T. Magkakasunod na thymidine adenosine. Okay? Then this one is your consensus strand. Initially, dito magbabind yung RNA polymerase mo. It's dalawang to. Okay? Then, with the help of your general transcription factor, i-guide niya si RNA polymerase dito sa dalawa na to. Once na mag-bind na si RNA polymerase dito sa promoter sequence mo or promoter site mo, okay? This is what you call the pre-initiation complex. Remember this. This is not part of your transcript, okay? Your data box, your consensus sequence, or generally your promoter will not be transcribed. It only tells you na malapit dito yung transcription area. Okay? When we say transcription area, when you go beyond that, before, that would be your upstream. Dito yan. When pag makarinig kayo ng upstream, downstream, that means left and right ng transcription site mo. Okay, this is your transcription site. Here is your upstream. Okay, before the sequences, before your transcription site is your upstream. Diyan normally makikita mo yung promoter mo. Kaya tinatawag ng mga negative. Negative 10, negative 35, negative 53. Okay? While from transcript site downward, that is your downstream. So mga +1, +2, +3, +4, +5. Your termination signals would take part on your downstream area. Okay? Direction tayo. Upstream, your promoters. Downstream, your termination signals and your elongation area. So, ulitin ko ha, promoter is not an RNA, it's not a DNA, it's not a protein, but a DNA sequence that is identified by your general transcription factors in eukaryotes for DNA-dependent RNA polymerase to initiate transcription. Doon siya magbabind para magkaroon ng pre-initiation complex. Now, this is on your tawag niyan, eukaryotes. Sa prokaryotes, may maririnig kayo na sigma factor. Si sigma factor yung pumapalit kay general transcription factors, okay, sa prokaryotes. Yung pinag-aralan nila noon was E. coli. So, sa E. coli, instead of general transcription factors, meron kang tinatawag na sigma factor. Okay, remember that. Now, when your RNA polymerase is already bound to your consensus sequence, that is your tawag niyan, close complex. Now, kung both na yan sila, or your promoter sequence is bound to your RNA polymerase, that's what we call open complex. Why? It stimulates to unwind. Na-unwind siya and nabre-break yung hydrogen bonds niya dito. Not by your helicase, but by your RNA polymerase. So, dalawa na yung trabaho ni RNA polymerase dito. Okay? It initiates transcription and it unwinds and destroys the hydrogen bonds. Kasi pagkatapos niyan, as the sequence progresses, binabalik niya rin yan. Okay? So, multi-functional si RNA polymerase dito. Unlike your DNA polymerase, paggawa lang talaga ng DNA template yung ginagawa niya. Pero si RNA polymerase, marami siyang trabaho dito. Okay? Now, how do you control the frequency of transcription? So, if there is bound on your GC box, ito ha, these are your promoters kasi. Ito yung CAAT box and GC box that are sequences na sunod-sunod, GC, or your CAAT box na bound to their protein, okay? SP1 and CTF complex. Yan silang dalawang complex, yan yung magle-label dun sa area na yon na ito yung inyong tawag niyan, i-transcribe more because kailangan pa nito sa system. Okay? Your GC box SP1 complex and your CAAT box CTF complex. Okay? They are the moderators. Okay? Pag nakabind dyan sa DNA area na yan, sa promoter area na yan, dyan magsisignal na kailangan more dito mag-transcribe na area na to. Kasi ito yung mas kailangan namin na protein. Yan yung gamit ni GC box SP1 complex and CAAT box CTF complex. Huwag malilito ha sa mga box. Now, yung summary ng this one, these are your general transcription factors. Sinamarize ko na lang kanina. Kasi this is the general picture para maka-initiate ka ng transcription. Kailangan itong lahat na protein na to and enzyme nakabind sa yung promoter area. Okay? This is your promoter area. Sino si enhancer? Okay? Si enhancer, yan yung mga tawag niyan, mga DNA sequence rin yan. It's a DNA sequence that will tell na ito siya, kailangan pang matranscribe more. Okay? Like your GC and your CAAT box. Those are your enhancers. Yan na yan. So, summary. Sobrang rami actually. Pero yung mga ni-mention ko, yan yung mga the usual. Now, sino si DNA dependent polymerase? Ang daming subunits nito that binds to magnesium ion and composes catalytic activity. Sinabi ko na kanina, it unwinds and dissolves hydrogen bond. Okay? Usually 20 base pairs. So, ang gamit nila lang talaga, okay, is for coding. Depende na lang sa mga i-code niya. RNA polymerase 2 codes for your MRNA, tapos small nucleus RNA, your silencing RNAs, while your polymerase 1 codes for your ribosomal RNA, while your RNA polymerase 3 codes for your transfer RNA or your ribosomal 5S ribosomal RNA. Please remember this, hindi lang si messenger RNA ang lumalabas sa transcription. Okay? It's not only messenger RNA that is produced, it also codes for any RNA that is needed in the system. Okay? Pero, ang pinakamaraming na kinako-code is messenger RNA kasi yung pinakakailangan talaga is the protein na maproproduce sa body. Another tawag niyan, yung full circle ng RNA transcription. Open complex formation, chain initiation, tapos tanggal na yung promoter clearance mo, then elongation. Ang pag-usapan natin next is your termination. Okay, kasi once na makastart na yan sila, naka-attach na sa promoter, naka-start na ng transcription, tuloy-tuloy yan. Okay? It takes a signal or a sequence. Kung may promoter sequence, meron tayong termination sequence. It takes another sequence to stop the transcription process. Okay? Zero factor is on your prokaryotes. Okay? This is a prokaryotic factor, zero factor. Please remember, sigma factor and rho factor are part of your prokaryotic transcription. Sa humans, dalawa yung ang ginagamitan natin ha. Ginagamit natin to stop the transcription. The C-terminal domain of your RNA polymerase and the cleavage and polyadenylation sequence. Ito yun. This one. Okay, this is the polyadenylation sequence. Tuloy-tuloy na A. Okay? Kasi meron rin tayong the CG sequence. Ito. It would form a hairpin, tapos magkakaroon sila ng temporary hydrogen bond. Pag maka-signal to sa system na, uy, meron tayo namang CGC na na sequence that created a hyphenated or a loop. Okay? It would take a pause. Okay? It would take a pause. Now, upon pausing, makikita niya yung yung kanya polyadenylated sequence. Diyan yung magsisignal kay RNA polymerase na this is the terminal sequence. Dito na ako magsa-stop. Okay? So yan. So initial transcript mo has a polyuracil tail. Okay? Initial transcript has a polyuracil trail. Okay? Remember that. After transcription, tapos na tayo sa transcription process, sinabi ko pa sa inyo, meron talagang mga walang gamit dyan. Mga sausaw suka, mga intro boys sa ating mRNA sequence na mRNA initial transcript. So, ang tawag dyan is introns. Okay? With the process called splicing by your spliceosome, diba? Nare-remember nyo si U1, U2, U4, U5, U6. The small nuclear RNAs, okay? They are the ones that do splicing. They do the post-transcription modification of your mRNA by removing the introns. Okay, this process here. Exon 1, codes for your protein yan. Okay? This one, sausaw suka, mga gap lang yan. Tatanggalin yan siya ng mga introns because they do not code a specific amino acid, your introns. Kaya yan sila pinatanggal. Kasi during translation, as we talk about translation later, tuloy-tuloy ang pag-translate ng mRNA mo to protein. Dapat walang pause. If ever man yung intron mo hindi na-splice or hindi natanggal dyan, there would be a pause on your translation. And that is a problem because it can cause mutation. Okay? Or furthermore, tawag niyan, automatic stop yung translation ng messenger RNA, incomplete protein. Okay, so that is the importance of your splicing because it can affect the translation process if it's not removed because it does not code for your any amino acids. Please remember that. So this one example is your beta thalassemia, inappropriate beta subunit of your hemoglobin, kaya nagkakaroon ka ng thalassemia. Pointless yung naproproduce niyang protein kasi hindi nakaka-function ng kanyang part. Now, another form of your post-transcription modification is adding 7-methylguanosine cap on the 5' end and polyaethyl and your 3' end. Okay? Importante rin to. Why? Your 7-methylguanosine cap would be the signal, okay, or the identification part of your messenger RNA na ito is for transcription. Okay? It would be identified by your ribosomal RNAs. While your polyaethyl, this one stabilizes your messenger RNA during translation. How? It binds as well to your ribosomal RNA, the coding one. Mamaya pag-uusapan natin during translation. So those two are important, ha? 7-methylguanosine identification, polyaethyl, that is your stabilization. during translation. And both is needed to facilitate translation and protect from exonucleases. Yung exonucleases, yan yung pwede makatanggal ng mga exons mo. Okay? And there are presence of microRNAs that tends to inhibit the translation of mRNA. Now, generally speaking, your ribosomal RNAs are transcripted and assembled at the nucleolus and transported to the ribosomes. As well as your transfer RNA is transcripted, and there is also post-modification to add this one. Okay? This is your AAC. This end is the one that attaches to your amino acid, specific amino acid. Okay, this is your anticodon loop. This is the one that attaches to your messenger RNA. This one identifies the specific. No, ito ba? Oh, ito. Ito yung nag-identify ng specific amino acid na ikakabit dito. Tapos ito, nagsastabilize siya. Pag-uusapan pa natin ito during translation. Now, protein synthesis and the genetic code. This is mainly translation process. Now, definition of terms. Codon. Please remember your codon is present in your messenger RNA. Hindi sa DNA, hindi sa transfer RNA, sa messenger RNA. Okay? Huwag malilito ha. Codon is present in your messenger RNA. How many nucleotides? Okay? 3. To have a codon, you need 3 nucleotides. Okay? 3 nucleotides. Because one codon represents one specific amino acid. Okay? That codon represents a specific amino acid. Pero, one amino acid can be coded by multiple codons. Remember that, ha? One codon, one amino acid. But one amino acid can be coded by multiple codons. So there are 3 codons that does not represent an amino acid. They are the known termination signals. Okay? Meron UGA, AOG, tapos UAG. Basta yan na yung tatlo na yan. When translation process starts, the mRNA would be continually read codon by codon until you stop on the termination signal. Okay? No punctuation. Kaya nga, importante ang splicing. Because, pag hindi mo ma-splice yung introns, magkakaroon ka ng punctuation. Diba? Masisira yung sequence mo. Other than that, baka automatic yun na spliceosome na yun, yung kailangan ma-splice would lead to coding of your termination signals. Masastop ka agad. Abnormal protein yung maproduce. There is no overlapping of reading of codons. Consequence per sequence tayo. And the genetic code is universal. Okay? There is definite representation of the organelles. Dapat meron, when we say universal kasi, kung anong kinukode ng DNA na to or ng DNA sequence na to, that is specific protein na talaga yan. Okay? Kahit saan kang DNA saang chromosome ka, yun ang i-code niya. Okay? So this is your tRNA. Please remember, your tRNA has the anticodon arm. This is the anticodon arm. This comprises 7 nucleotides, pero yung anticodon mo, of course, 3 lang. Sa mRNA, doon mo makikita si codon. So AAA ang lalabas. Every arm has a function. Minention ko na kanina. So recognition and attachment. the specific amino acid to be placed in your tRNA is recognized by your D-arm. Okay? Your ribothymin pseudouridine cytidine arm, ito siya. Yan. It binds to your bosomal surface for stabilization. And sinabi ko nga, your anticodon arm has 7 nucleotides and they are read in 3' to 5'. Your messenger RNA is read 5' to 3'. Your transfer RNA is read 3' to 5'. Now, anong gamit ni amino acyl transferase? Synthase. This is your amino acid, specific amino acid. Here is your amino acid, tRNA synthase, and with the help of your ATP, it would produce this complex. Your enzyme, adenosine, and your amino acid. Okay? Now, with the help still of your amino acyl tRNA synthetase, it would react on your distal arm, dito siya, sa acceptor arm, kakabit dyan. See? Ito siya. Ikakabit ni amino acyl tRNA synthase, and after that, lalabas na si adenosine monophosphate with the enzyme. Tuloy-tuloy yan yung kanilang function. And please remember, one amino acyl tRNA synthetase acts on one tRNA. Okay, very specific, or one amino acid. Sorry, one amino acid. So, kung meron kang 20 amino acid, then you have 20 specific amino acyl tRNA synthetase. Okay? Because very specific itong enzyme na to. 1:1 lang talaga. Now, what do you call the wobble effect? Si wobble effect, ito yung parang glitch in a good way ng ng system. So this one here, it can be coded if ever your codons has the same code for the amino acid, the anticodon can bind to that codon kahit hindi sila magka-base pair. Okay? Example dito, usually yung nagdi-dictate naman kasi ng specificity ng codon is the third nucleotide to be coded. Dito siya. Okay? So, UCC and UCU codes for one amino acid. So, kahit C pa yan ang andito or U pa yan, same. Okay, anticodon yung magkakabit dyan, same amino acid. So, walang problema. That is your wobble effect. It suggests that the base pairing of the last nucleotide may not follow the Watson-Crick pairing na AUCG, pwedeng UG. Okay? Example, your arginine codons, AGA and AGG, can bind the same anticodon having the uracil on the 5' end. Now, na natin yung process, okay, ng pagkakabit, pagta-transcribe. Hindi transcribe, pagta-translate. So, na-identify na natin yung amino acid na kakabit. What if nagka-problema during transcription? Okay? What if may problema sa translation? It would produce, paulit-ulit kung sinasabi kanina, mutations. We have the so-called single base mutation or point mutation. Either transition or transversion. So, when we say transition, kung sabihin natin, pyrimidine, purine to purine. Okay? Napalitan lang yan sila. Okay? Of the same kind. Pyrimidine to pyrimidine exchange or purine to purine exchange. Pero sa transversion, yung pyrimidine dapat nagiging purine. Or yung purine dapat naging pyrimidine. Pero isang nucleotide lang yan. That's what we call point mutation. Okay? Pwede makaproduce ito ng silent mutation. Kung silent mutation, walang problema. Kasi yung codon na yun na nabago, same amino acid yung kinocode niya. So makakaproduce siya ng silent mutation. Walang problema. Paano kung missense mutation or missense effect? Ibang amino acid yung maproproduce. Imbis na glutamate, nagiging valine. Diba? Meron tayo niyan. Those are sa ano yan? Sa HBS natin, hemoglobin S. Those are missense mutation. Kasi dapat glutamine nagiging valine. Now, meron naman tinatawag na nonsense mutation. Pag sabihin nonsense mutation, yung na-code niya is your terminal codon. Okay? Or your stop codon. That means hindi complete yung protein na na-produce sa nonsense mutation. Okay, please remember the difference between a silent mutation, a missense mutation, and a nonsense mutation. So, acceptable missense, partially acceptable missense, and unacceptable missense. It is based on the clinical picture of the patient. Okay? Pagsabihan natin na acceptable missense, almost same functional group pa or functional unit yung napalit na amino acid, like your hemoglobin A, beta chain, and hemoglobin hickory. So on the 61 position na amino acid, the lysine nagkaroon ng missense mutation, naging asparagine. Okay, almost same functional group, so walang problema. So hindi siya severe yung symptoms. We have the partially acceptable missense. Yun na nga yung sinabi ko sa inyo na hemoglobin S. On the 6th position, si glutamate napalitan ng valine. So ang nangyayari, yung structural abnormality ng hemoglobin, pag deoxidize, nagsisikel siya. That means abnormality na siya na nakaka-produce ng disease talaga. Then lastly, your unacceptable missense. This is not a nonsense, ha? This is a missense mutation. Okay? It still codes for your amino acid. It does not code for a stop codon. So on the 58th position, histidine is replaced by your tyrosine. Mga example lang to, that would produce hemoglobin M or your Boston. Hemoglobin Boston. Dito, severe anemia ang pinoproduce and can cause death. Okay? Severe mutation talaga to. Now, we have, second, kung may point mutation, meron tayong frame shift mutation. There is an altered reading frame of your mRNA due to deletion or insertion of a single or multiple nucleotides. Okay? Kung natanggalan man or hindi man natanggalan ng introns, it can cause frame shift mutations and can produce major alterations of amino acid sequence, absence of multiple amino acids, or even nonsense codon and end prematurely. This is the kinds of your frame shift mutations. This is the normal. Okay? This is the normal. Pag maglagay ka dyan ng isang amino acid, mapapalitan na to ng coding. Okay? Itong lahat na to, iba na ang coding. Kasi instead of CUU, magiging UCU, UCG, AAA, GGC, UAU. Yan yung example. So when we say garbled, lahat niyan mapapalitan na amino acid kasi lahat tawag niyan, lahat may iba yung coding kasi we read, di ba, 3 nucleotides ang nagko-compose ng codon. So magdagdag ka dyan ng isa na nucleotide lang, mapapalitan na yung sequence of nucleotides. Okay? Now, deletion pala to. Pagtanggal ka naman ng tatlo, if you remove 3 nucleotides, so 1 codon is removed, that would dictate 1 amino acid removed. Okay? Hindi masyadong garbled yung inyong sequence. Okay? Because specific yung ito siya. Specific lang to sa isang amino acid. Unlike dito, lahat niyan masisira yung coding. Now, kung dito tayo sa insertion, magdagdag ka ng isa, magagarble yan. So, masisira yung coding mo dyan. Then, pwede kang magkaroon ng code for premature stopping. Paano ko naman kung may insertion and deletion? Napagtripan ka ni God. So, dito, magdadagdag ka ng uracil, may babawasan ka ng cytosine. So, pwede itong makakode ng iba. This area lang ang garbled. Okay? Kasi, ang mangyayari dito, parang manu-neutralize yung addition. So, nag-add ka naman dito, pero nabawasan naman dito. So, ito lang na area yung mag-iiba ng amino acid sequence. Okay? Pero yung the rest na hindi naapektuhan ng addition pati insertion, walang problema. Okay? Ito yung mga mas grabe yung effect ng mga frame shift mutations. Unlike your point mutations, konti lang by a single protein or a single amino acid. Dito talaga buong amino acid sequence mo masisira. Now, doon na tayo sa translation proper, the protein synthesis. It has a lot of same as to your transcription, meron tayong initiation, may elongation, meron tayong termination. Let's talk about initiation. It has first there should be a dissociation of your ribosome. Di ba meron 70S, di ba yun? Magiging 40S and 60S subunits. Maghihiwalay sila. Ipapakita yung picture later. Okay? Now, there would be a binding of your ternary complex to the 40S ribosome to form the 43S pre-initiation complex. Your ternary ang ternary complex is this one. Initiation complex, initiator methionine tRNA, eukaryotic initiation factor 2, and your GTP. Ito silang tatlo yung nagko-compose ng ternary complex. Please remember these three, ha? Okay? An RNA, a protein, and then a nucleotide, your guanosine triphosphate. Sila yung nagko-compose ng ternary complex mo. Okay? Then, after that, after na naghiwalay ng 70S ribosome mo, then yung ternary complex mo nakabind na sa 40S mo, your mRNA will bind to your pre-initiation complex. Okay, sa 40 pa rin. So, from 43S, given na yung nakakakabit na yung mRNA mo, would produce your 48S pre-initiation complex. After that, mag-fuse ulit silang dalawa. Di ba, sineparate natin to 40S and 60S subunit, tapos kabit tayo ng kabit dun sa 40S ternary complex and your messenger RNA nakakabit dun, i-fuse niya ulit yan to produce an 80S initiation complex. We will, ipapakita ko sa inyo later. I just want you to remember these steps. Dissociate first, binding of the ternary complex, binding of the messenger RNA, then fusion back. So, na-mention natin yung ito siya, eukaryotic initiation factor 2. Marami pa yan sila actually. There is a broad group of eukaryotic initiation factors. So, I enumerated each, yung mga importante lang, ha? So, eukaryotic initiation factor 3 and eukaryotic initiation factor 1A, it binds to your 40S subunit. Okay? It is not part of your tertiary complex. Separate lang yan sila. It binds to your 40S subunit kasi it inhibits fusion back of your 40S and 60S by binding to your 40S subunit. Number 2, your EIF 4F. 4F is a complex, okay, composed of your 4E, 4G, and 4A complex. It is the cap binding protein complex. Okay? Di ba sabi ko sa inyo, yung guanosine cap ni 5' mRNA, para siya makilala, kailangan mo ng EIF4F complex. Okay? Your eukaryotic initiation factor 4F. So the 4E part recognizes and attaches to the cap of your mRNA. Please remember this is the rate limiting step of your translation. The recognition of the cap of your mRNA by the EIF4E. Okay? Now, your 4G naman, si 4G, nagbabind siya kay initiation factor 3. Okay? And to the poly-A tail binding proteins. Anong gamit ni poly-A tail binding proteins? Ito siya, yan yung mga bridge mo. So, meron kang 4G, meron kang poly-A tail binding protein, meron ka ng poly-A tail. Kasi, di ba, yung function ng poly-A tail mo is stability during translation. Your cap is for your recognition, your poly-A tail is for stability. Okay? And the one that attaches to your poly-A tail is your poly-A tail binding proteins, and the one that attaches to your poly-A tail binding proteins is your 4G. Saan niya kinakabit? Sa 40S subunit. Okay? Through IF3. Okay? Laro to ng mga bridge. So, papakita ko sa inyo yun ng mga bridges na yan. Pero dito muna tayo. Third is eukaryotic initiation factor 4B. It melts the secondary structure of your mRNA. Okay? Then your EIF5, hydrolysis of your GTP and EIF2 complex, that would lead to the fusion of your 48S subunit and 60S subunit to form 80S ribosome. So it would act here on the number 4. So this is acted by your eukaryotic initiation factor 5. Okay? Ito na yan, yung sinasabi ko sa inyo. So in summary, di ba, initiation natin, separate muna 40S, 60S. Then your tertiary complex, it would bind to your 40S. Okay? 40S. Then, yung initiation factor 3 mo and 1A, dito siya pa rin magbabind sa 40S para hindi sila mag-fuse. Then, meron tayo yung 4F complex mo. This one. This 4F complex will bind to your poly-A binding proteins. Ito siya. Ito siyang nakahilera na to. This is your poly-A tail. Because this complex will stabilize your translation process. And sabi ko kanina, si 4E would recognize the guanosine cap, methyl guanosine cap. Ito siya. So nakikita nyo, parang circle siya. And after that, naka-set up na to. Naka-set up na to siya. And na-recognize na dito. Usually, methionine yan ang initial. So, magbabind siya with the help of your initiation factor 5. Okay? That's the function of your initiation factor 5 for this to become this one. Tapos, tuloy-tuloy na yan. That is the process. Tapos, pag makakita siya ng stop codon, this is AAU, diyan na siya magsa-stop. Tapos, magdi-dissociate na naman. Then, kung meron na naman bagong messenger RNA na na-trap, ulit tong system na to. Perpetual ang trabaho nila actually, but different messenger RNAs because they would code for different proteins. Remember the functions of eukaryotic initiation factor. So, how do we regulate? Okay? How do we regulate the initiation? Because, sabi ko pa nga, this is the rate limiting step. Your 4E, identifying your messenger RNA and binding to it on the cap is the rate limiting step of translation. So, usually merong nakakabit kay tawag niyan, eukaryotic initiation factor 4E. It inhibits its activity. Okay, that is 4E binding protein. Madali lang ma-memorize. Ang nakakabit kay EIF4E, si 4E binding protein. Okay? With the help of your insulin and your polypeptide, mitogenic polypeptides and growth factors, it would promote phosphorylation, okay, of 4E binding protein. It would be deactivated, si 4EBP. Once deactivated, it would be removed kay EIF4E, then active na si EIF4E. Once activated na si EIF4E, it would form your 4F complex with 4G and 4A. Ito siya. Okay? Kailangan nitong complex na to identify the cap. So kung may presence ng 4E binding protein, hindi activated ang 4E mo, wala kang 4F complex, hindi matutuloy yung translation mo. So importanteng ma-phosphorylate or ma-deactivate lang muna si 4E binding protein para maka-form ng 4F complex para ma-identify si cap. Kasi pag hindi talaga ma-identify si cap, hindi makaka-initiate yung translation. Okay? Kumbaga, tawag ito? Barangay. Kung di rin iyo kilala kapitan, di rin ka mo makakapapirmahin medical certificate. The barangay medical certificate. Pero, para niyo makilala kapitan, kailangan niyon secretary. That is the 4F. Maraming secretary si CAP. Okay, secretary number 1, secretary 2, na secretary number 3. Okay, that are your 4F. That's the analogy. Okay, so importante, the lesson here, importante ang secretary sa barangay hall. Kasi doon mo lang makikilala si CAP. Now, tapos na tayo sa initiation. Rate limiting factor, determining the cap. Sa elongation process naman, 4 din. Same as to your initiation. So, the binding of your amino acyl tRNA to the A site. Okay? Automatic na yan ha. Huwag kakalimutan. Automatic na yan na si tRNA mo napupunta dun sa A site, meron na talagang amino acid. Hindi pa siya labas-pasok. Automatic, meron na siyang dala. Okay? To the A site. It is guided by your GTP and your elongation factor 1A. Second, the peptide bond formation or the transfer of the amino acid to another tRNA, amino acyl tRNA. Then translocation of the ribosome to the mRNA. So mag-shift siya kasi tapos na yung codon ng first codon ng mRNA na code na. Second codon, pag-move yan, that is your translocation. And the expulsion of the deacylated tRNA or yung tRNA na wala ng amino acid from the P side, okay, from the polypeptide side to your exit side. Ito, makinig. Kasi importante. Si peptidyl transferase, that is your component of the 28 sRNA of the 60 subunit dito. Okay? It attacks the amino acid peptide bond. Ito siya. Saan ako dito? Yan, dito. So, i-attack niya yan, ikakabit niya to dito. Then, kailangan mo mag-move yan kasi. So, your transfer RNA here would transfer to your exit, then remove. Then, the one carrying the 2 amino acids or your one peptide bond, magta-transfer sa peptide area. Okay? Because A is for acceptance, P is for peptide, E is for exit. Okay? Yung sinabi ko pa sa inyo na maraming factors sa sa translation. We have your elongation factor 2. Ang nagmo-move ng transfer RNA from here to here is your elongation factor 2. It displaces the peptidyl tRNA from the A site to the P site, while the deacylated tRNA is on the, ito siya, deacylated naman to, wala na tong amino acid, to the exit site. Tapos lalabas siya. Okay? Then same process. While one codon, specific tRNA with the specific amino acid, kakabit. Then your peptidyl transferase or your ribozymes would cleave this one and bind it to here. Then, once na bound na siya, your elongation factor 2 would move this deacylated transfer RNA to the exit site. Then your peptide tRNA would be moved to your peptide site. Tapos lalabas, then repeat. Paulit-ulit yan hanggang makode si stop codon. Pag may stop codon na dyan, tapos na tong process na to. So, that is the process of elongation. Okay? Automatic na yan. Pag start pa lang, huwag niyong kakalimutan. Pag start pa lang, meron na dyan sa peptide area. Yung initial mo, yung initial protein mo, usually methionine yan, na initial amino acid na nakakabit dyan. Okay? Now, energy requirement. Baka tanongin sa iyo kasi favorite ni Dr. Monsanto yung ilang ATP, ilang highly active phosphate ang ginagamit sa mga ganito, sa ganyan. So, to produce a peptide bond, one peptide bond, isa lang to, you at least need 4 high energy phosphates. Saan yan galing na 4 high energy phosphates? First, there would be a hydrolysis of your ATP to adenosine monophosphate for the amino acid binding to tRNA. Okay? That is 2 na. They're automatic, dalawa na kaagad yan na high energy phosphate. Tapos, yung dalawang GTP na nagiging GTP para makapasok si amino acyl transferase o transfer RNA sa protein, that's 1. And second, na GTP na kakailanganin mo is for transfer from your active site or your acceptance site to your peptide site. Okay? So, tanungin kayo, Dr. Monsanto, ilang high energy phosphates ang kailangan to produce one peptide bond? It would take 4. Okay? It would take 4. Tapos na tayo sa elongation. Okay? Sa elongation, nakita natin yung transition from your acceptance to your peptide to your exit with the help of your elongation factor 2 and with the help of your peptidyl transferase enzyme. Sa termination, simple lang, stop codon. Okay? Sa stop codon, walang tRNA na nakaka-identify nito. Okay? Once pumasok si stop codon sa ribosomal RNA mo, walang kakabit na transfer RNA. Walang pupunta sa acceptance site dito. Wala. Kasi stop codon siya eh. That means walang amino acid na dapat andyan. Okay? Now, we have the releasing factors. Okay, those are proteins that help to identify your stop codon. Remember this, no transfer RNA that identifies your stop codon. It would be your releasing factor 1 and releasing factor 3 that would identify your stop codon. Now, anong ginagawa nila? With the help of GTP and peptidyltransferase, it promotes hydrolysis between the peptide and the tRNA in the P site. Ito siya. Yan yung kanyang ipopromote. Your RF, your releasing factor. Releasing factor, bibitawan pa para mabitawan na niya yan dyan. Okay? With the help pa rin of your peptidyltransferase. Okay? And upon release of the protein and the tRNA in the P site, tapos mag-dissociate na siya. Balik na ulit dito. This one. Kasi may stop codon na detect na siya ng iyong releasing factor 1 and releasing factor 3. Natanggal na yung newly synthesized polypeptide chain mo, then magdi-dissociate na yung ATS mo. Now, sino si polysome, sino si P-bodies? Now, polysomes are multiple ribosomes translating one messenger RNA. Okay, group of ribosomes that will translate single mRNA, usually separated between 35 nucleotides minimum. Yan. Kasi given the fact na malalaki yan sila na RNAs, ang ribosomal RNAs, kailangan at least 35 nucleotides ang gap. And usually, polysomes, pag nag-group, yan yung nakikita niya via electron microscope na rough picture ng endoplasmic reticulum. Okay? Due to your polysomes. Sino naman si P-bodies? Storage factor ito ng storage mechanism to ng system mo for your mRNAs. Okay? Once na nasa cytoplasm to, yung mRNA na produce during transcription na hindi mo pa kailangan i-translate, doon mo siya i-store sa P-bodies mo. Minsan doon na rin siya na-dissolve. Okay, sa P-bodies. Storage and dissolution ng messenger RNA mo, nasa P-bodies yan. Now, what are the antibiotics used in the market that inhibits protein synthesis in the bacteria? We have your tetracyclines, remember? It binds to your amino acyl tRNA to the A site of the ribosome. So, kung sino man na amino acyl tRNA yung nasa A site, hindi na siya makukode. Okay? Kaya magkakaroon ng hindi makaka-produce ng protein kasi blocked yung A site mo. The A site would be inhibited. So, wala nang papasok na bagong amino acid, hindi nakahaba yung protein ng bacteria, mamamatay siya. Yan yung function ng tetracyclines mo. Your chloramphenicol and macrolides naman inhibit the 23S ribosomal RNA of your 60S subunit. Your puromycin is a structural analog of your tyrosinyl tRNA. It inhibits the ribosomes, both eukaryotic and prokaryotic inhibition. Cyclohexamide inhibits ribosomes in the eukaryotic cell. And your diphtheria toxin inactivates elongation factor 2. Please remember this. What's the function of your diphtheria toxin? Inactivates elongation factor 2. So, kung walang elongation factor 2, walang transfer of RNA o peptide bond, walang transfer RNA from your P site to the exit, from your acceptance site to your P site. Okay? Kaya importante itong diphtheria toxin na to. Tatanungin kayo nito, internal medicine, pag second year ninyo, the internal medicine, microbiology, pediatrics, paulit-ulit itong itatanong sa iyo. What is the function of your diphtheria toxin? It inactivates elongation factor 2. aneurysine, inactivation of the 28S ribosomal RNA. I will not dig deep dito sa mga antibiotics na to because may pharmacology kayo nito. Doon na kayo papahirapan nito. Sa biochem, sa ano lang muna tayo, sa basic na transcription translation. Last chapter for this afternoon. How do we regulate gene expression? Two types, positive regulation, negative regulation. When we say positive regulation, it is promoted by your enhancer or activator. That means consistently ito siya na proteins na ma-define. Okay? The more ito matra-transcribe, the more ito matra-translate. That is positive regulation. Vice versa, negative regulation, silencer or repressor, it would diminish your expression. Okay, so decrease protein synthesis of that specific protein. Now, there are three types of response to regulators. Meron kang regulators na dyan, may it be a positive or a negative regulator. Yung body natin, we have three responses. The type A, na kung andyan lang yung regulator or nagtatrabaho yung regulator mo or nagproproduce siya ng signal, dun lang magre-respond or mag-increase ng gene expression. Okay? Pag walang activity si regulator mo, wala rin, normal lang, average lang. Pero pag present na naman siya, balik na naman, taas yung gene expression mo. Dito, magaling lang sa simula, type B. Okay? At the start of the signal, magpipik siya. Then eventually, would decrease even with the presence of the signal. So may particular time lang sa start ng pag-produce ng signal na yon na doon lang siya tataas. Pero after that specific time, kahit andyan pa yung signal, tuloy babalik siya sa normal. Then would come a recovery phase. Okay, nawala yung signal, ganun pa rin. Then another signal na naman ang magpo-produce, magpipik siya konti, tapos babalik na naman. That is your type B. Only transient increase. Usually, ito siya yung tinatawag na antibiotics. Sa mga medications natin, ganito yung expression ng antibiotic yung response ng body natin sa antibiotic. Initial lang muna nagre-respond, tapos pagkalaunan na, kahit ilang beses mo nang ibigay, nawawala na yung effect. Pero kung mag-take a pause ka lang muna, hayaan mo after 1 to 2 years, hindi ka mag-take nitong antibiotic, magbigay ka na naman, magiging effective na naman siya. Basta hindi lang long periods of time ka mag-antibiotic with the same antibiotic. Okay? Makakaroon ka ng type B or transient increase lang. Type C, okay, once lang. Mag-signal ka lang ng isang beses, tuloy-tuloy na. Kahit mawala yung signal, tuloy-tuloy pa rin yung trabaho. Okay? Ito yung frustrating ko sa Harpers. Ang hirap kasi sa Harpers, pinaghahalo-halo niya yung prokaryotic and eukaryotic. So, sometimes may human, sometimes may bacteria. Biology pa naman rin sila. Pero, yun nga, yung ginamit nilang basis for study of humans is the study of, of bacteria. Specifically, E. coli. Yun yung kanilang well-studied na na subject sa biochemistry, especially on genetics. So here, we have the prokaryotic gene expression. Now, definition of terms na naman. Ito yung mahirap. Kailangan marami kang i-memorize na mga kung sino-sino. Si operon, this one, it is a linear array of genes. Okay, it's not a gene but a group of genes, si operon, that takes part in a specific metabolic pathway. Okay, like your lactose metabolism, ito, example to nila, your lactose metabolism is a metabolic pathway. So this is the operon for lactose metabolism. Okay? This is the operon for your lactose metabolism. Sa prokaryotic, cistron is the smallest unit of gene expression. Now, when we say polycistronic mRNA, sa sa prokaryotes, ang cistron yung nagko-code ng specific protein. Polycistronic mRNA, it is a single mRNA that codes for more than one protein. Sa prokaryotic lang yan ha, because sa atin, one messenger RNA, one protein. Sa bacteria, pwedeng magkaroon ng one mRNA, more than one protein ang i-code. That is your polycistronic messenger RNA. Then we also have your inducible gene. We have this inducible gene is its expression is increased in response to an inducer or activator. So, given the fact na kailangan niya ng inducer or activator to increase gene expression, automatic natin may isip na there is low basal rate of transcription. That means special lang siyang tina-transcribe for specific for special purposes, not daily basis. Okay? Not your muscle proteins. Muscle proteins wear and tear, always yan na-produce. Pero mga specific actions ng body or mga rare functions ng body, doon lang siya magko-code si inducible gene. So, if there is an inducer or an activator present in response to a specific stimuli, doon lang siya i-produce. Okay? Now, meron tayong tinatawag na housekeeping gene. It is expressed in a constant rate. Then we have also your catabolic repression phenomenon. Paano to ma-describe na catabolic repression phenomenon? Meron kang glucose and lactose. Okay? Kinain mo pareho. Initially, glucose talaga yung unang i-metabolize niyan. Due to the presence of glucose, it can inhibit your cyclic adenyl cyclase. Pag may inhibit si cyclic adenyl cyclase, hindi ma-produce si cyclic adenosine monophosphate. Pag wala si cyclic adenosine monophosphate, hindi magbabind yung mRNA mo sa ribosomal RNA. Okay? So, kung may presence ng glucose, may inhibit yung expression ng lactose operon. Okay? Because walang camp na maproproduce. I-elaborate ko sa next slide to. Then we have your operator locus. Area of the double-stranded DNA that has a high affinity with the repressor proteins. Then your gratuitous inducer. It is an analog of the primary stimuli. Kung baka copycat lang, hindi naman copycat, analog or almost kapareho lang ng itsura ng primary stimuli mo, pero same effect lang. Okay? Parang tawag nito? Doc Monsanto, Doktora Casal. Okay? Halos hindi na naman sila halos magkapareho lang sila, pero same respect pa rin yung binibigay mo. That is your gratuitous inducer. Yung sinasabi ko kanina, yung example ng catabolite repression phenomenon, yung lactose operon may inhibit kasi with the presence of your glucose, because your glucose or glycerol inhibit your adenyl cyclase. Pag inhibited si adenyl cyclase, wala kang maproproduce na camp. Pag wala kang camp, walang magbabind to your catabolite gene complex. Okay? Pag walang camp complex, sa prokaryotic lang to ha, sa E. coli to, pag walang camp complex, hindi maproproduce yung pre-initiation complex mo sa translation. Okay? Yan yung function niya. These are mga specific phenomenons na on gene expression, pero well studied lang to sa E. coli. Pero baka lang itanong ni Dr. Bonsanto sa inyong finals. Your catabolite repression phenomenon. Ito. Another studied body in your biochemistry ni Harper's, your bacteriophage lambda. Importante kasi ito sila because it provided the groundwork or the foundation of what you are eukaryotic studies today. Si bacteriophage, it is a virus for your bacteria, di ba? Astig. Bacteriophage is a virus for your bacteria. Now, meron tayong tinatawag na bacteriophage lambda. Okay, this is the studied bacteriophage before. It states that normally it has two phases, the lysogenic and the lytic pathway. Okay, it depends on the environment and the gene involved. Okay, it depends on the environment and the gene involved. Ang nag-ano nito, ang nagko-control kasi dito na identify ng mga scientists yung molecular switch. Okay? Your molecular switch happens when there would be a change of environment or a change of stimuli. So, sa bacteriophage, sa kanyang DNA sequence, there is tinatawag na right operator locus. Okay? Sino si right operator locus? It is the one that codes for the that codes for the phases of your bacteriophage lambda. Kung meron kang poor nutritional state, your bacteriophage would code for your CI gene. Okay, yan yung matra-transcript, yan yung matra-translate. Once it is transcripted and translated, it would produce a CI protein. Si CI protein would then eventually bind to your CI gene and a specific areas of your operator locus, OR1 and OR2, to prevent transcription of your CRO gene. So sa lysogenic pathway wherein there is poor nutritional state, CI gene ma-activate, CRO gene deactivated. Okay? Anong nangyayari naman dito? Dormant state to ng bacteriophage mo. Hindi siya magre-replicate because the transcription is inhibited by your CI protein coded by your CI gene. Now, paano kung may dangerous na signal or DNA damaging signal or rich nutritional state? Ang mangyayari, magkocode na naman si right operator locus. Magkocode siya. Ang i-code niya si CRO gene. Now, si CRO gene would produce a CRO protein that would bind to your CRO gene and to your operator 3, OR3 gene. Dito siya, ang kanyang two functions would be inhibit the CI repressor gene and further transcription of your CRO gene. Once there is further transcription of your CRO gene, doon na magre-replicate yung DNA ni bacteriophage lambda. Once there is further replication of the DNA of the bacteriophage, makakaproduce siya ng 100 copies of the completely packaged virus, then magra-rupture yung bacteria mo kasi sa sobrang rami. Okay? That happens to your lytic pathway. In summary, ng ating bacteriophage lambda na part, lysogenic pathway, your CI gene is activated that produces CI protein that would inhibit your CRO gene. It is present during a poor nutritional state and so that there would be integration. Okay, integration only of your bacteriophage DNA sa DNA ng bacteria. That is during lysogenic pathway. Pero kung merong DNA damaging signals na maproproduce or meron kang rich nutritional state, ma-activate yung lytic pathway wherein your CRO gene is activated that would produce CRO protein. Okay? Then eventually, DNA replication would commence to produce 100 copies of completely packaged virus, then leads to the lysis of the bacterium. Yan, ito yan siya. Yung pinagnangawak-ngawak ko kanina. This is your lytic phase. This is your lysogenic phase. For nutritional state, walang magagamit for DNA replication. So, yan, mag-fuse siya. And active dyan is your CI gene. Pero dito, sa lytic phase, kung may DNA damaging signals, i-activate niya si REC-A hydrolase and would promote CRO-MRNA o CRO promoter. Okay? Your CRO gene. So yan. Nakikita nyo? Repressor mRNA or your CI repressor and your CRO promoter or your CRO gene. Lytic phase, lysogenic phase. Tandaan nyo lang yan. Kasi lilito-lituhin kayo dyan. During lysogenic phase, anong gene ang present? Or anong gene ang activated? Yan. Examples lang yan ang pwedeng i-test sa inyo. So, enumerated here are the special regulation of eukaryotic gene. We have the differential expression. Merong areas na may gamit talaga. May areas din na hindi siya gamit. So, usually, inhibited yung kanyang expression. Minsan, kung doon naman sa kailangan siya ng area, doon lang siya ipropromote, okay? Na ano, for example, your beta peptide or your beta chain sa hemoglobin, sa bone marrow, kailangan siya. So, actively transcribed and translated siya sa bone marrow. Pero kung sa muscle naman, wala namang gamit si beta chains sa muscle mo. Okay? Wala siyang gamit doon, so hindi siya na-transcribe or translate sa muscles. That is differential expression. Depende lang kung saang area siya gamit. Now, we have the histone covalent modification. This is one elaborated modification ng histone because histone, aside from stabilizing your DNA and creating nucleosome, it affects it can detect the transcription and replication of your DNA, your histone. So, your histone covalent modification or the histone code, quote unquote, is due to your post-translational modifications. Post-translational modifications? Akala ko post-transcription. Okay? When we say post-translational modifications, this happens to proteins. Okay? When you have histones that have special functions, histones are proteins, please remember that. If you have proteins that have special functions, they undergo post-translational modifications. Now, may tatlo yan na proteins na gumagawa niyan or enzymes. The code writers, these are the one that promotes post-translational modifications. Now, pag tapos na yung post-translational modification, it would be acted upon by your code readers. Those are proteins or enzymes that recognize and binds to your post-translational modification areas. Code erasers, pag tapos na yung function niya, tatanggalin na niya yung post-translational modification. So these are the writers, readers, and erasers. Ipapakita ko next slide. Now, methylation of deoxycysteine residues. Diba? Na-mention ko, sa active zone, they are less methylated. Pero sa inactive, okay, so para hindi siya makode masyado, mag-add ka ng methyl doon sa deoxycysteine residues. Please remember this, kung ano yung minimethylate to produce active and inactive zone. Please remember, cysteine residues yan ha, deoxycysteine residues, yung minimethylate to produce active and inactive zones. Nalagay ko na to sa previous slide ko sa first discussion natin. These are the kinuha ko lang. Possible roles of modified histones. Okay, I discussed that already. Lastly, the last topic for this afternoon, epigenetic mechanisms. So, what is epigenetics? It is a study of chemical modifications and or molecules that alter gene expression patterns. Okay, kung gaano ka kadalas, okay, kung gaano kadalas siya i-transcript and translate, okay, without altering the underlying DNA sequence. We have two forms of your epigenetic signals. Your transsignal, it is a signal that is coded and produced by a specific gene, usually protein kasi ito ng mga signal natin. Coded by your specific gene, and it can go in and out of the cell. Okay? It can go in and out of the cell. After cell division, okay, magse-separate, separate cytoplasm, separate nucleus. Yung preformed signal na nasama during cell division, papasok ulit siya sa nucleus. Initially, lumabas siya. Tapos after cell division, papasok ulit siya. Then it would bind to its own area. Okay, its own gene sequence. It would bind there and it would promote production. Okay, it would promote production of its own. Parang self-regulatory lang to sila. Ang pagkakaiba lang kay cis, nakakabit na kagad. Okay, hindi siya in and out of the cell. Automatic kabit siya. Upon upon initiation. So ito siya. This is your trans-epigenetic signal. So this is the gene that codes for this one. Okay. Now it can go in and out of the cell. Then once there would be cell division, of course there would be a division of cytoplasm. Some would go to the first, some would go to the second. Then it would re-enter the nucleus and would stimulate production of its own. Okay, that is trans-epigenetic signal. What is cis? Cis is, wala siyang pakialam sa kanyang own production. Ang kanyang ginagawa lang is that, initially, cis is, andyan na yan siya talaga. Pero, yung kanyang pinopromote is not her own. Okay? A different protein. So, during nucleus division, yan, the sister chromatid or sister chromosome would code for another signal. Okay? Para meron siyang sarili. Okay? Kasi hindi mo siya pwedeng i-transfer. So, ang gagawin niya, total, andito naman yung code, gagawa na lang siya ng kanyang sarili. Tapos ikakabit niya sa kanyang sarili para makaproduce siya ng kanyang sariling specific protein. Kasi kung wala tong cis signal na to, tanggalin mo yan, okay, sabihin natin natanggal yan, hindi siya makakaproduce ng protein na to. Kung ano man yung hindi niya mapropromote yung transcription nito. So mawawalan siya ng protein. Kasi importante itong cis epigenetic signal mo. Okay? Required to siyang protein na to or signal na to so that this area can be transcribed. Unlike here na nakaproduce na siya on his own, Tapos, babalik na lang siya para stimulate ulit yung sa kanya. Dito, during the process pa lang of cell division, inside the nucleus, diretso na siya nagpo-produce. Okay? That is trans-epigenetic signal and your cis-epigenetic signal. Okay, same cell lang sila nanggagaling sa cis. Si trans, from the initial cell siya. Now, enhancers na mention ko na sa inyo. Distal sequences sa double-stranded DNA that would promote transcription of the particular dsNA sequence. Okay? So these are the characteristics of the properties of enhancers. It can work when located long distance from the promoter or the promoter region. It can work upstream or downstream, +1, -1 on the promoter. Tapos either direction niya, pwede niyang ma-stimulate for transcription. It can promote homologous or heterologous it can work with homologous or heterologous promoters. It can work by binding one or more protein. It can stimulate for the dsNA, the double-stranded DNA na makaka-attach yung specific proteins doon. Tapos it can work recruiting chromatin modifying co-regulatory complexes. And it can work by facilitating binding of the basal transcription complex to cis-link promoter. Okay, very deep na to, pero ang gusto ko lang makuha yung idea is that enhancers are sequences in the same double-stranded DNA na would promote transcription of particular double-stranded DNA sequence, not its own but a separate sequence. Okay. So we have the enhancer. Okay, we have the enhancer. Let's talk about the protein binding of the regulatory proteins. When we say motifs, kung ano yung kanyang itsura pag kumakabit siya. Okay, kung ano yung kanyang process ng pagkabit. These are the general ideas. I'll just enumerate this. It has a high affinity to a specific site and low affinity to other DNA sequence. Okay? Kung saan siya kailangan, doon siya ma-attract, never sa iba. Loyal. Now, a region of the protein is bound to the DNA while other surfaces can be bound to your other regulatory factors. Okay? Kailangan may free part siya for other regulatory factors, pero definitely nakakabit siya sa DNA. Now, your protein-DNA interaction is based on hydrogen bonds, ionic interactions, and van der Waals forces. Fourth, motifs found in protein is class-specific. Okay? That means when we say na this protein is for DNA only. Okay? Kung ano man yung nasa DNA mo na protein, para sa kanya lang yan. Wala nang iba. Wala siyang ibang function kundi to regulate the DNA. Okay? Kung meron kang protein na nakita doon na malapit sa DNA, na unknown yung trabaho, it would suggest that the protein binds to a DNA. Okay? And dito, proteins with helix turn helix or leucine zipper motifs form dimers, and their respective DNA binding sites are symmetric palindromes. Okay, palindromes, papakita ko sa inyo yung anong itsura ng idea ng palindrome. Para bang kasal ng kapatid ko, yung kanyang date ng kasal niya is 02/02/2020. February 2, 2020 yung kanyang kasal. So either way mo siya basahin, same lang ang kanyang itsura, 02/02/2020. Okay? Same as kung babasahin mo siya pabaliktad, ganun pa rin, 02/02/2020. For zinc finger motifs, it binds binding site repeat 2 to 9 times. Papakita ko yung mga itsura kasi ito parang in summary ito yung itsura lang talaga ng pag-bind ng protein to, yung kanyang motif. So these are the examples of your motifs na present. First is your helix turn helix motif. Okay? It is the first motif to be identified, and each monomer is composed of 3 antiparallel beta sheets. Okay? This is a monomer ha. One monomer to. Please remember this ha. One monomer. Okay? One monomer pa lang yan. Anong general natin? It forms a dimer. Now, paano siya magkakaroon ng dimer? If one here is connected to a major group and one here is connected to a major group, it would be connected by your antiparallel beta sheets. Ito siya. Tabang ko lang to. Ito siya. One monomer containing 3 alpha helices. Yan siya. Connected by antiparallel. Pag sabihin natin antiparallel, so this one, this one, this one, this one. These are beta sheets, ha? Okay? Ito yung magkakabit from one monomer to another monomer. Okay? That is your helix turn helix motif. Zinc finger naman, si zinc containing proteins, tapos meron kang cysteine and histidine. Okay? These are your fingers. Remember the amino acid. Cysteine and histidine fingers. It binds to major groups. Okay? It binds to major groups. Hindi siya sumusuksok. Unlike your helix turn helix. Yan. Helix turn helix. Dito siya, outside lang talaga siya, and mga kanyang galamay na lang yung kumakabit dyan. Okay? This is one protein, ha? Please remember that this is one protein. And lastly, your leucine zipper motif forms an alpha helix with a leucine repeat on every 7th position. Okay? It occurs on 8 helix turns and 4 protein repeats. Your 2 monomers may zip together in a coil and form a tight dimeric complex. Okay? Ito yan siya. One monomer. Tapos ito yan siya, yan yung zipper mo. Okay? This is yung tinatawag na zipper. Leucine zipper motif. This one are your leucines. Siya yung nagbabind dyan na area. Kaya tinatawag na ang kanyang itsura is leucine zipper. Okay? This is your leucine zipper motif. Top view lang to. Ito side view. So, yan lang yung mga motif mo. Mga itsura niya kung paano siya nagbabind. Kasi quote and quote, sabi ng mga scientists, it can affect their function or the regulation that they do for the DNA. Tapos na tayo. Okay. We're done with the chapter 36, 37, and 38. 39 would be your reading assignment. I don't know when is your schedule for the quiz. Tatanungin ko pa kay Bossing Monsanto. Pero I'll give you 5 minutes to review kasi I will test your short-term memory and long-term memory. Okay? This is non-graded. This is just to tawag niyan? This is just to bind your knowledge na natutunan kung meron man ngayong hapon. And magre-request lang ako. This is the last last meeting na natin for this semester. Magre-request lang ako, each one of 120 kayo, no? 120 kayong tinuruan ko. First impression and things that I can improve during on my teaching. Isang na lang ninyo, kahit naka-code name, walang problema. Kung matapang at tao ka, pwedeng full name mo isulat mo doon sa ano mo, sa evaluation sa akin. Okay? So, walang problema 'yon. I take criticism. Bad, good, nonsense, okay lang. So, please do that and submit to me this 6 p.m. I-ano na lang, i-message na lang sa akin. Sinong sekretary ninyo? Siya na lang ano, tawag niyan, magsosolicit sa inyo. Tapos, put it on a Word document or PDF, submit na lang sa akin later. Yun na lang yung i-request ko sa inyo. Kasi para next batch, kung magturo pa ako ng biochem, tingnan ko lang. At least alam ko yung aking short end and long end of the stick. Okay? 5 minutes, review the topic. Kung yung mga notes ninyo, then let's have a short quiz. The usual, 10 to 20. Okay? Okay. Doesn't make you dance when you jump with your friends at a party. What's your favorite song? Doesn't make you smile till you think of me. Your eyes tell me, what are you dreaming? Everything I wanna know at all. I spent 10,000 hours, 10,000 more. But that's what it takes to learn that sweetheart of yours. And I might never